Okay, so let's start with hopefully the last part of our lecture today, cloning from single cell to whole animal. Again, bear in mind this is theoretical, right? So how do we do it? Well, the first step is you have to grow a complete animal from single cell which has one copy of the gene. That is the rule. Secondly, you have to breed the animal with other heterozygous animal to ensure that the gene is stably integrated. And C, you have to test the offspring, whereby each offspring has to have at least 25% chance of being homologous for the donor gene. Okay, so let's take a look at this example of Dolly. So cloning of Dolly, how it is done. So we know that Dolly was created in 1996, even though it says 1997 because it was when it was announced to the whole world. So target cells to be cloned were obtained from pregnant ewes udder. So the female sheep is called ewes. So the cells were then non-embryonic cell involved in adult cell cloning. Cells were cultured under nutrient medium and induced to inactivity. This is to ensure that lots of embryos could be obtained from it. An unfertilized egg was obtained from another ewe, so the second one, whereby the nucleus is removed because we don't want the property to be passed on to the clone animal, and in this case Dolly. So both cells were fused via electric pulse, and division took place many times after many tries, not in one try. The embryo was then planted after about 6 days to another ewe, which is the surrogate mother. So with normal gestation, Dolly was born. So everything is quite normal actually for Dolly. And this is how you or the researcher needs to do to make sure that the founder animals, it has the gene stably integrated into it. So the breeding, production of transgenic mice utilize almost all of the methods, but for larger animals there is a bit of an exception, and of course, as it says here, harder to create. So as it says, to date only microinjection and nuclear transfer have been used to create livestock. So this is how you do the breeding. So I hope everyone still remembers these logos, especially those who have learned on genetics as MBB. So this symbol signifies for female, this symbol signifies for male. If it has a color in it, means that it is affected, or whatever the test or whatever it signifies, which means that it is affected. Half, which means that it is half affected or maybe a carrier. So when you mate a male and female, it will produce the generation. This is the offspring, and when you mate the offspring, you produce the next generation. So this is F1, F2, and F3. So as you can see over here, what the breeding of the pedigree chart tells us is that, so founder animals over here, we know that it is not fully integrated with the genes, so that's why we put it as half. So it is mated with another animal, but which doesn't carry the same gene. So when it produces the offspring, this is by the Mendelian genetics, at least one of it, at least 25% of the offspring carries the transgene, or 25% integration of the homozygous gene. So you made this homozygous of the offspring with another homozygous of another transgenic animal, in the hope that this is by the Mendelian genetic as well, whereby you get 1:1 integration. So at least one first fully homozygous offspring, and of course, usually it doesn't really stop there. You need to make sure that it does carry both alleles or fully homozygous to make sure that the gene is stably integrated. So there is an explanation over here. First generation of offspring is the founder animals. Second generation, F2, produced from transgenic and non-transgenic parent. And the third generation is obtained from both transgenic parents and will usually have more stable genes, not probably fully integrated, but more stable than the parent. So if you have any queries so far on the breeding, just jot it down and post. I will actually open up a forum where you can post a question, or we can discuss it a bit on Tuesday. So I'm going to proceed with the next topic, which is on bST, or bovine somatotrophin. So bovine, as the name says, comes from cattle, bulls, cows, and so forth. So these are a topic on growth hormone. So somatotrophin are growth hormone found only in cattle. So somatotrophins are protein growth hormone. Okay, they are species specific in their action and effect. Why we are discussing on somatotrophin? It has something to do with us. Okay, in a while. So cows, humans, pigs, mice, and other organisms produce their own unique somatotrophin. So the hormones actually they are not cross-active, okay, and because they are produced by different animals, they are unique, and to each usually they have a very signifies or synchronized function, okay, but for each animal, okay, usually okay, they are either lactose sensitive or basically there are some sensitivity involved over here. So bST is produced by pituitary gland at the base of the cattle's brain. The same goes with human actually. So important for growth, development, and other bodily function, but usually again is for growth hormone. Injecting bST into lactating cows, okay, over the years have been found to increase milk production, and this was discovered way earlier in 1930s. So how did they do that back in 1930s? They have a way of doing it. You can just search on the history. It's too long to say over here. So scientists have since then determined which gene in cattle codes for bST production. And of course, because they have known for that, they removed the gene and insert it into E. coli to manipulate it in vitro. So they purified the bST produced from the background and injected it back into the cattle to increase the production of the milk. So more or less, this is how bST is being produced. So generally, sorry for that, my cats again. This is how bST is being produced. Okay. The original bST has been identified, more or less, okay, cloned into E. coli, and then fermented, upscaled, okay, and then inserted it back into the cow after it has been purified and isolated. So, what is the correlation of bST and milk production? So, bST must be injected into the cattle on a regular basis for effective milk production. It's about 2 to 3 weeks interval. So bST will be broken down immediately into small inactive amino acid and peptides once it has been ingested. So frequency of injecting bST will depend on whether bST product can be developed for longer time or not. And this is where it is unique from one animal to another because it depends. Prior to injection of bST, milk yield are found to be increased. This is in cow and in other animals they haven't actually tested it yet. Cattle, sorry. So the hypothesis of this rationale to isolate bST and then inject it back into lactating cows is to because of the postulation that blood flow to mammary gland is increased by injecting more genes. Blood, as we know, carries increased amount of nutrient, so the mammary gland will extract more nutrient to improve milk production, and hence they postulate or they hypothesize that more milk is going to be produced, thus increase the efficiency of feed and decreasing the use of feed for body maintenance. But in actual, after they have done all this, they have done all the testing and see how it actually functions and the behavior of the animal. More nutrients are taken in by the mammary gland for bloodstream, to synthesize more milk. So one of the hypotheses is actually half true. bST will affect the synthesis and secretion of insulin-like growth factor (IGFI). To what extent it hasn't been reported and exposed yet. Cow increases the feed intake because more nutrients are needed to take basically to synthesize more milk. So but they found that there is an increased efficiency in conversion of feed to milk due to unchanged nutrient requirement for body function. So more or less, I would summarize it as such that it is a successful test, a successful method to increase milk production. So milk composition between treated and untreated cows with bST supplementation actually does not differ, but the yield differ. The one that has been treated with bST, there is an increase in milk production or yield. So in terms of us human consuming the cow's milk or the cattle milk, so everyone here drink milk, right? Whether when you are small or now. So humans break down both types of bST, whether natural or the ones that has been incorporated into the inactive product, okay, by prior to digestion without a problem. So that means that there is no problem with us, and it has been tested, even for those with lactose intolerant. So there is no problem at all. But okay, uh, probably those with lactose intolerant may want to be careful about it as well. Okay, you may have stomach discomfort somehow. So using this method, milk production was found to be increased about 5 to 15% in the US, France, Germany, Netherlands, and UK, the country which produces the milk the most and export the milk to all over the world. So what are the benefits and the risk of bST? Of course, everything has its pros and cons. So the benefits are the milk contains the same amount of injected bST. So, but in terms of milk production, is it increased? Biological activity of commercial bST is actually identical to naturally produced bST. So there is no problem with the gene that it could be expressed and it functions properly. So both bST, normal and also the clone ones, is immediately broken down into inactive amino acid and peptides in the human digestive tract. So that means that there would be no discomfort for us. bST technology, however, may decline cost for milk production. Should be milk cost, or the price of milk should be decreased, but we actually haven't seen it yet. So hopefully in the future we will. Fingers crossed. So the risk. What are the risks? So it has been found that cows that have been injected with bST have longer calving period, which means that they do not reach reproduction stage for about 2 to 4 weeks up until 6 weeks. So longer calving intervals. They remain teens. And not to say they remain as teens. So they do not want to engage to reproduce. So they have also lower conception rates, and high doses increase death rate of calf embryos. So farmers and also scientists have to be careful not to inject too much into the cow for fear of killing the animal in the process unintentionally. Now, what are the limitations of this technique? So it could not be used in cows with less than 100 days into lactation. So that means that cows which is in advanced lactation stage producing milk couldn't be injected with more bST. It could not be used in cows with chronic health or fertility problems for fear of endangering or compromising the life, and it is expected to be used less in heifers than adult cows. So basically heifers, which means that young cows, so the expectation to be used is actually lower, so because it needs to be at the right stage, a bit more mature stage. So that is more or less the introduction on how the gene could be cloned in the lab and the benefit to the milk industry. So I'm going to proceed with transgenic mice a bit. So what I'm going to touch about transgenic mice is just the basic stuff. More advanced stuff is going to be discussed later on, probably either on Tuesday if we have the time, or next week on Sunday. So this is the core, or the more content of transgenic animal is based on transgenic mice. So it is actually one of the tools to explore many biological questions, because we know that mice is it is more or less kind of like a model animals, and to a certain extent people say that they have similar genome to human. How similar is our genome to the mice? We'll see in a while. So the best thing about mice and why they are being used as model animal is because they reproduce rapidly, have short lifespans, they are inexpensive, they are actually quite cheap, and can be genetically manipulated easily. So mice and humans have roughly the same number of nucleotides in their genome, hence people more or less postulate that we have similar genomes. And in comparing genes, both human and mice have similar genes, but with different structure and activities of the protein products. That is to be expected. So these are the comparative of genome size of humans and other organisms that has been studied. So humans, Homo sapiens, are us, with rat, mouse. There is a difference between rat and mouse. For those of you who didn't know, or you can Google up to see on the feature. There is a difference even in the feature. Comparing to fruit fly, Arabidopsis thaliana, a plant, the model plant, roundworm, C. elegans, yeast, C. cerevisiae, S. cerevisiae, and bacteria, E. coli, and also some bacteria, influenza bacteria. So as you can see, okay, the chromosome number is more or less similar, 46 to 42. Yeah. Size is, of course, again, not that much difference between us and them. Okay. Also, estimated number of genes that has been identified and whatnot. And this is taking a look at the karyotype of similarities between mouse and human genetics. So this, as you can see, it's the chromosomes. They only have up until, they only have 42 chromosomes. Okay, one is sex. We only have 22 plus one sex chromosomes. Okay, but the location of some of the genes in specified chromosomes are similar. So more or less that is just the general about transgenic mice that I'm going to discuss for this Sunday. So on this diagram, again, actually it would be better if we were face to face, but never mind. I'll just say it out loud. Usually I have a questions for students to guess what does all this H1, C, G, and O means. So this is from chromosome 1, 2, and 3, and these are from animals which are said to be or to have genomes which are similar to humans. If anyone could guess, then very good. Okay, so H stands for human, us. C stands for, anyone can guess? Okay, never mind. G and O. Okay, so G is basically for gorilla. O is for orangutan. So C, I'll leave it for you to guess. And for those of you who wants to guess, okay, we can discuss on Tuesday. Alright, let's leave it a bit straight over here. So as you can see, okay, from the 3 chromosomes, okay, the size, okay, and genes location, okay, within the chromosomes are more or less quite similar, right? Okay, so the organism of choice for mammalian genetic engineers, especially for human in terms of medical field, is actually mice as well. It is easily manipulated because of the small size. It is hardy. Hardy means that, doesn't mean that it's hard in structure, which means that it could withstand a lot of condition, okay, and test, and easy for lots of manipulation. It has short life cycle. This is actually important so that we do not cause more sufferings to them. So genetically, it is possible because it has been more or less compared with human, and a lot of useful strains and tools are available to study in genetic mice, okay, and for comparison of human and mice transgene and also genome. So before we move on to the third activity, if you have any queries, any question, okay, you can post it down there underneath the video, or you can email me, or I will open, like I said, I will open up a forum. You can post your question in a forum. So your third activity before we break off for today is on puzzles. Do you like puzzles? Alright, so of course individual activity, since we are all very far apart, find as many words as you can in puzzles. So the words that you need to find has to be related to function of transgenic mice, not just any word, not any word that you think is English or Malay or whatever, but it has to be related to function of transgenic mice, okay? So don't just give me on, off, or whatever, or cat, as you can see over here. Cat, no, it's not related to transgenic mice. Yes, cat hunts mice, but it's not related to function of transgenic mice, okay? So we will have a discussion on activity 2 and activity 3 on Tuesday. So get ready with your activity. So I'll just leave this for a while for you to, well, screen capture or basically take a shot, okay, with your handphone and do your activity later on. But I would say I would end my lecture now, and well, let's meet on Tuesday via Zoom. So I would require for everyone to, well, download Zoom either or install Zoom in your computer, your laptop, or your handphone. If anyone has any problems with the installation or your internet access, just let me know through WhatsApp or through emails So that probably I will change it to Google Meet, because since that seems that everyone has Gmail account, so probably it'll be easier. Like I said, the other method is via a live Instagram, but I'm actually not too prone on that since I don't have Facebook. But let's see. I'm willing to try on Zoom first. I've used it for a while now, okay, with meetings, so it works fine. So let's try. And everyone, please download and install Zoom, ZOOM, in your handphone or your laptop. Okay, till then, we will meet on Tuesday. Bye bye.